I'm just going to introduce our team members to start off with. I am Amanda Foster. This is wife Ferrari, sorry. Emily, Erica M, Erica S, Ailey, Beth, April, and Hannah.
All right, good morning everybody. We are the iGEM team from Missouri University of Science and Technology, the premier university in the Midwest for science and engineering. Our school mascot is called Joe Minor, and so we decided to use the iGEM logo to come up with our own mascot, Joe Microbe. All right, and our team was started at Missouri S&T in 2007. We've come a long way since then in our small beginnings in the last 4 years. This has been especially evident in the past year. Last fall, we became a recognized student organization, and that gave us legitimate funding within the school and also very much presence in the student body. Last fall, we also won the award for Missouri S&T's Most Improved Student Organization. A very big step for us in that we were being recognized for our quick jump from being a very small, little-known organization to very much a presence in the school. This year, we're going to be taking another big step as well. Our school currently has 14 award-winning design teams, and that is very much a source of pride and identity for our school, and it gives students hands-on experience within science-related fields, and also gives them a chance to present their ideas in competitions. We've currently submitted our application for Student Design and Experiential Learning Center to become an official design team for the school. We're very excited for this opportunity and that we are very much going to be able to expand upon the design teams that our school currently has. No other design team does anything near synthetic biology like what we do, and so we very much hope that by joining the design teams we can diversify the very prestigious ranks of Missouri S&T. Now, with how far we've come in the past 4 years, we're very excited to present to you our newest project, the glucose sensor.
In a healthy human body, the pancreas responds to increases in glucose concentration via specialized cells known as pancreatic islets. These cells, as shown in the diagram above, produce insulin in response to increasing glucose concentrations. The insulin is then released into the blood, and as you can see in the upper left-hand portion of the diagram, when the insulin is detected by receptors in the cell membrane, it induces the cellular uptake of glucose. The glucose can then be used immediately for energy or stored in a different form for a later date. In people with type 1 diabetes, the pancreas or the cells in the pancreas responsible for the production of insulin have been damaged or destroyed by the body's own immune system. This means that they are no longer capable of producing the necessary amounts of insulin that required to maintain a healthy body. In type 2 diabetes, the pancreas is still capable of producing the necessary amounts of insulin. However, due to a diminished response of the pancreas in response to increasing glucose concentrations, it produces the inadequate amount of insulin. Current tools being used to manage this condition include insulin pumps and glucose sensors, both of which can be unavailable or unpractical for lower income populations, and in the case of insulin pumps, can be inefficient. Our team wanted to develop an alternative solution that was more economically sound and that utilized synthetic biology. We found that the 2007 Type A iGEM team had developed a system that sensed increases in glucose concentration or changes in osmolarity, and in response to these changes in osmolarity, fluoresced. However, the team did not submit a BioBrit part for this system. Our team decided to pick up where the Type A 2007 team left off with our current project.
It is estimated that over 3 million people in the U.S. alone are affected by type 1 diabetes. The American Diabetes Association also estimates that the diagnosis and treatment of diabetes costs Americans over $200 billion per year. Complications caused by diabetes include heart disease, stroke, high blood pressure, blindness, kidney disease, and neuropathy, but many of these can be prevented by testing blood glucose levels and administering insulin shots at appropriate times. The goal of this project is to create and mutate a glucose sensor to produce different mutations that can detect various glucose concentrations. This research could lead to the creation of cheaper blood glucose testing devices and could potentially aid in the development of more practical insulin pump technology.
To begin our project, our team started with 3 different parts from the iGEM registry. First, the RBS3, a medium-strength ribosome binding site. We also used the EYFP gene, which codes for yellow fluorescence protein, and the OMP-C operator, which contains 3 binding sites for phosphorylated OMP-R. OMP-R is a constituent of the ENVZ OMP-R 2-component regulatory system. In order to complete our part, we utilized a series of restriction digests and ligations. We first cut open the EYFP gene from its plasmid and inserted it downstream of the RBS. This gave us our first intermediate part, which was submitted to the registry. We then cut open this intermediate part and inserted it downstream of the OMP-C operator, and this gave us our final part. This is our team's favorite part, and it was characterized through testing.
Now that you know a little bit about our part, I'm going to introduce you to the 2-part regulatory system that we use. In this system, there's the ENVZ intermembrane protein that senses a change in osmolarity. Once this osmolarity change is sensed, it will phosphorylate OMP-R, and then the OMP-R can undergo cooperative binding to the OMP-R binding sites. When sites 1 and 2 are bound, the RNAP can transcribe downstream, turning on the EYFP gene, and the system will fluoresce. But however, when the 3rd site is bound, it blocks the RNAP binding site, and it can no longer transcribe downstream and turn on the EYFP gene, and our system is off. So in summary, when there's low to moderate levels of osmolarity, the system will be activated. But when there's high levels of osmolarity, the system will downregulate fluorescence.
Activation of our gene was measured by utilizing the fluorescence reporter gene. A 96-well plate reader was used to measure fluorescence. Our cultures were grown in half concentration LB and were diluted in a series so that the concentration of glucose levels would remain constant. Cultures were taken from each treatment and were read and were imaged with a 96-well plate reader, and data was collected.
We use this testing to characterize our part through sequence verification, dose response, and time trials. Our parts were sequence verified, and these are the fluorescence wavelengths that we used for our assays. We grew our part in the E. coli strain DH5 alpha under standard conditions and in liquid cultures. We decided to use liquid cultures because testing the changes in osmolarity was a lot easier in a liquid culture than on a solid plate. We also found that the LB media does not work well with our system because even without additional glucose, the osmolarity of the LB is high enough to downregulate the system when any additional glucose is added. Here's the graph of our dose response with the high osmolarity LB media. As you can see, after the 0% background, with each increasing level of glucose, the system fluoresces less and less, so it's a negative regulation of the system. So we decided to use half concentrated LB, and so this is our dose response with half concentrated LB. As you can see, from the 0% background to the 1%, there's a significant increase in fluorescence, and then after that, with increasing concentrations of glucose, there's the negative feedback begins after the 1% threshold. So in summary, we used the half concentrated LBs so that we could observe positive regulation from 0 to 1% glucose and then negative regulation after the 1% threshold.
Our team also tested our part over time. This graph shows 0%, 1%, and 8% glucose over 4.5-hour time span. Due to the lag time between adding glucose to our cells and imaging our plate, our 0-hour time point is actually around 20 minutes. But as you can see, Around 20 minutes gives a good induction of the system. Also seen in this graph, like Amanda said, 1% glucose gives a higher induction over 0% glucose, and 8% glucose induces downregulation of the system. Another significant part of our project was mutagenesis. The OMP-R binding region could be mutated so that the binding affinity of the OMP-R protein to the DNA strand was altered. Theoretically, this altered binding affinity would give the system the ability to be sensitive to glucose at different concentrations. Our team used a mutator strain E. coli to perform mutagenesis. We followed standard procedures, or standard protocol. At first, our team plated possible mutants on glucose plates, but due to the difficulty of quantifying fluorescence on plates, we soon inoculated liquid cultures and screened for fluorescence in liquid cultures. After weeks of testing, though, our team did not find any mutants. Although, we do believe that with more time, we can find a sufficient amount of mutations to represent a comprehensive glucose concentration scale. Once these mutations were isolated and characterized, they could be applied to a new glucose sensing system. The fluorescence reporter gene could be removed and replaced with a reporter gene that is easier to visualize. For example, we could use a color reporter gene to represent different glucose concentrations. We propose that we use the Cambridge 2009 team's eChromite project for this purpose. The Cambridge team developed 4 reporter genes that are very good indicators. Our project uses the 2007 Type A team's ideas and the 2009 Cambridge team's eChromite project to build simple, affordable glucose test strips. Test strips that could provide basic alternatives to lower income populations. Our system also lays the groundwork for future development and research into new insulin pump technology.
I'm going to talk to you guys about the educational events that we did, since our research in synthetic biology, has the potential to affect the community, and as our campus and town in rural Missouri are on the conservative side, we felt it would be important to educate the public. We held a synthetic biology informational for students and faculty. We told them what synthetic biology is and what iGEM does. We talked about past projects and then asked the audience about other applications and other project ideas, and we were really pleased with the responses that they gave us. We then developed a class to help our team mature. It's a class through the university called Biological Innovation and Design. Our goals were to teach students standard molecular genetics techniques and develop project proposals. We extensively researched 4 topics and came up with project proposals for those, and then we chose a project from those 4. It organized our team, but it also left a lasting effect on the university because this class will be offered each spring. We took 2 project proposals to the undergraduate research conference, which is campus-wide, and we won first place with one of our projects. We exposed iGEM to students, faculty, but also the vice provost of undergraduate studies, and he seemed really impressed with us. We believe this networking helped us win the Most Improved Organization Award. We developed an educational activity to teach kids about synthetic biology. We performed this at the local high school and on campus when the leadership group came to visit us, and both groups were really engaged in the activity and seemed to enjoy it. We collaborated with leadership and cultural programs on an open forum they host called Speak Up, Speak Out. We suggested the topic of synthetic biology and then participated in the discussion. The organization said that it was the best discussion they had all semester. With these educational events, it was really rewarding to teach people about synthetic biology and then to receive a positive response and interest in what we do. This was positive exposure for our team, but iGEM as a whole as well.
We'd also like to give a big thank you finally to our wonderful advisors who with that none of this would have been possible if they hadn't been there to help us out and guide us through the process as well. Dr. David Westenberg and Dr. Katie Shannon. We'd also like to thank some of our mentors for our group, Vanessa Kagan and Daniel Rausch. Vanessa helped us with sequencing our DNA, and Daniel was the former president of iGEM, and he helped mentor new members and also to guide us through the process as well. And also we'd like to thank some of our friends in the lab who were unable to join us today. Our lab manager, David Pullman, has put in so much time and effort into this project, and also Lou Harmon, who was our webmaster and designed our wiki. And we would like to give a final thank you to our sponsors for providing us such awesome financial support, the Missouri S&T Department of Biological Sciences, Chemical Engineering, Chemistry, Student Council, and Office of Undergraduate Studies, ExxonMobil, and Photo9. Now, thank you very much for being a great audience for us, and we would like to open up the floor for questions.
Thank you very much. And I love your logo. It's awesome. All right. So I have a question about. I was really impressed with the consideration of the concentration of solutes in your growth media, and how you address that problem by cutting back the concentration of it so it wouldn't affect your system. I do have a question about the expected function of the promoter. So if I understand this correctly, high concentrations of glucose should repress the promoter. So I'm a little unclear about your proposed application of using this to be activated by glucose, and then also the proposal to mutate it so that it's, I'm assuming, less sensitive to glucose, so you can get a range of output. But wouldn't that just create a constitutive promoter that's always on when you're right? So I guess I'm unclear about the proposal to make this so that it's actually turned on by glucose. Is that actually possible?
The idea is to have several different mutants in the glucose sensor that have different threshold values of fluorescence. So the highest fluorescence would be observed or if we used a color reporter, then the color would be highest at the different thresholds. So if we have 4 different mutants, each one would have a different color, so you could see which one was expressed the most, and that would correspond to the concentration. Does that answer your question?
Almost. So I guess I'll just wrap that up as a comment for the sake of time, but it seems that the function of the promoter is to be repressed in the presence of glucose. And I'm not quite seeing how it could be mutated or altered to actually come up in the presence of glucose instead. And then I'll come to your poster to talk about possible ways to sort of, because this is a very promising lead up to something, if you were to introduce an inverter perhaps into the system rather than just the promoter directly. But I'll talk to you about that.
If you want to see me after the presentation, I actually have an answer for that.
Okay, thank you.
So can you tell me a little bit more about choosing osmolarity as your sort of measure of glucose? How indirect a measurement is that?
It is an indirect measurement. We chose to work with the osmolarity sensor because the Type A team had already developed a project plan for this, and we just had to pick up where they left off and finish it up. Also, through our background research, our team was not able to find a more direct system of sensing glucose that was an easily attainable goal. Does that answer your question?
Yes, thank you.
So I have a question regarding the graph you show on the time trials at 0%, 1%, and 8% glucose. So what's missing is this graph is a control of the background. So I want to, have you ever done the background DHY alpha? What's the curve look like?
Yes, we did do a background DH5 alpha. The background fluorescence is right around the 8% glucose. Okay. We also have blanks on our dose responses that show the background if you would like to see them. Yes?
Could you go back to the graph? I think that was right before that.
This is the dose response with the half concentrated LB.
Okay. So it did go up 1%. I just wanted to make sure that.
You mentioned that you had a couple educational events and described them briefly in the presentation and on your wiki. A couple of questions on the content of the exchanges that took place. You said that they were constructive and that you felt great coming out of them. One of them was a public event, and you mentioned the somewhat conservative cultural context within which you live and work. What issues came up at that event on synthetic biology and its relationship to that local culture?
At the events themselves, there weren't really issues. Just within our community, people are really nervous when you say the words genetic engineering together. So we were more trying to fight that by starting with children and telling them what exactly we're doing and the risks and also the benefits, because we recognize there are risks and we want to show them there are also really big benefits.
And you also mentioned that you had a speak up, speak out session with a university audience. And the background of the people in the audience was somewhat limited in terms of biology. To what extent were you able to get good questions and good dialogue going on horizontal gene transfer and E. coli, or on any of the real safety issues that people do talk about with your general audience?
Is this specific to the Speak Up, Speak Out?
Either speak up, speak out, or in general. Because you're a technical group, you're interacting with populations or audiences that don't necessarily have a lot of technical background. And how did you overcome, if you will, that basic barrier?
Well, we started small with kind of like basic demonstrations and figures that represented genes. We constructed a Lego plasmid and showed them like taking genes in and out and how synthetic biology works. So we focused more for this first year of really trying to contact the community. We're focusing more on really, really basic concepts. So what synthetic biology is, what bioengineering really means. And we don't really get into the details, the nitty-gritty of it, because we didn't think that they would be as receptive.
Sure.
And does that answer your question?
Yeah.
The leadership group who hosts Speak Up, Speak Out also prepared an example, like a couple of different research example scenarios of how synthetic biology can have both positive and negative effects depending on the application, but we didn't really get into the specifics of the safety issues.
Well, we'll talk more maybe at poster session as well, but I'm going to open it to the audience if there are questions or comments. I know it's always awkward when you have the teams that are following sitting in the audience. You want to talk about the definition of a mutual hostage situation. Comments or questions from the audience? Further questions from the judges?
Do we want to go back to the
Yeah, why don't you go
first
back
question?
Do you mind repeating your question again?
Okay, so I guess to make that a little bit more clear, so I would just like some clarity on sort of what characteristic of the promoter makes it so that when you do mutate it, it's going to respond, it's going to be activated at higher levels of glucose.
So if you The system can be, next one, oh, sorry, I'm going to graphs. The system can be characterized, what's the word, that contrast, or just dose responses in general. Okay, can be calibrated for different sensitivities of glucose. The range of glucose that we'd be looking for in the human blood is actually below 1%. So if we test below 1%, we would get a straight increase. We wouldn't even have to worry about downregulation unless the medium we're testing in has a high osmolarity. And we do realize that blood has a high osmolarity, so something in the application would have to be done to clean up the sample that we were testing, or we could calibrate the system. So you're worried about downregulation of the system?
So my question is based on the biology of the promoter. Yes. It seems that there's an off switch. I don't understand how the on works, so repressors are binding the promoter.
We would have to.
What makes it actually come up?
What makes it come up is when when you have a low or moderate amount of osmolarity, so not too overpowering, when you have a low or moderate amount of osmolarity and 1 OMPAR binds, you're going to get transcription. With higher amounts of osmolarity, the binding frequency is going to increase.
So when you have the 1 OMPAR binding, is that actually increasing transcription?
Yes,
above the basal?
Yes. When we have lower concentrations where we only get 1 or 2 OMPAR binding to the promoter, the phosphorylated OMPAR recruits RNA polymerase to transcribe the gene.
That that makes it a lot clearer, and I didn't quite catch that.
Sorry.
On the wiki or in the thank you.
And as you increase osmolarity until you reach that overpowering threshold, you're going to get a positive correlation in transcription.